So, hello everyone. Good afternoon. Welcome to the 2024 Sahyadri Outstanding Plant Postdoc Symposium. Just a reminder that this symposium is being recorded, and I'm gonna share my screen now to introduce a little bit about our event today. I am Israel Cunha-Neto. I am a postdoc at New York University and a member of the leadership team for Plant Postdocs. And for those who don't know, Plant Postdocs is a community of plant science postdocs helping each other to reach the next career stage. So we are present on social media. You can find us through email, but also on Blue Sky, on X. We have a thriving community on Slack. You are welcome to join us and communicate and be part of this group. We also have our YouTube channel where we have most of our past symposia and webinar. And this symposium will be also hosted on our YouTube channel. So keep in touch with us, interact with our community, and be part of this group of postdocs. Before we give the floor to our speakers today, I have one announcement about some of the Plant Postdocs activities, and that is about something that we are launching very soon, which is a new area on our website that's called Plant Postdocs Database. In this area, we have the 3PRD, which is something that some of you might be part of already. That is the Potential Plant Peer Reviewer Database, a place where you can log in your information and volunteer to be contacted by editors to do some peer reviewer work. So if you're interested in getting some experience on that, that's a place for you to interact with our community. And a new resource in that area is the P2PSD, the Potential Plant Speaker Database. That is a place for you to network with our colleagues if you are interested in having more opportunities to share about your research, but also outreach activities, and that could be through seminars, through conference, and other activities. So stay tuned to our social media. We will soon announce officially the launch of this new resource on our website. All right. I'm honored to moderate the second Sahyadri Symposium today, and this symposium features the presentations of the awards recipients. This award is named after the Western Ghats mountain range, South India. Sahyadri refers to the benevolent mountains and is one of the biodiversity hotspots in the world. This award was established in 2023. It was created to highlight exceptional postdocs in plant-related fields who are navigating the academic job search in the US. So each awardee was paired with a mentor who helped them through this job search, and we are cheering for their success in this challenging journey. So please join me in congratulating the 2024 Sahyadri Outstanding Plant Postdocs, Dr. Deepak, Dr. Renee Dale, and Dr. Sombir Rao. They're going to have about 12 to 15 minutes to talk about their research. The order of presentations follows what we have in the screen right now. Dr. Deepak is the first presenter. He is a postdoctoral researcher at Michigan State University. His research focuses on plant microbe interactions and the logistics of plant defense. So today his talk is titled Linking Secretion and Cytoskeleton in Plant Immunity. So please take the floor, Dr. Deepak, or better take the screen. You can share now your screen. And for those who are attending the symposium, we will have time also for questions. So please write your questions on the chat and make sure to pass them along to the speakers.
So thank you so much, Israel, for the introduction. And I also wanted to take this opportunity to thank the Plant Postdocs for this immense honor. This award is special to my heart because I come from the south of India, and Western Ghats are very close to home. And for those who have not visited it, please go visit it. It's not only a great biodiversity hotspot. In my opinion, it has one of the best coffees in the world. Right? So as Israel mentioned, my research interest lies in plant microbe interactions. And what I want to understand in my academic career is how plants manage the logistics of defense, how they are able to put things where they're supposed to be, and what are the transportation pathways that they take. A different research interest of mine is also to understand how plant pathogen interactions shape cell wall fortification, which helps plants to prepare for the next set of battles coming their way. Right. So today I'm going to present a part of my postdoc work, which is linking the secretion and the cytoskeleton, especially in plant immunity against bacterial pathogens. And all of this will be from a perspective of a new protein that we have discovered called trans-Golgi network associated protein 1. Right. So perfect. So one thing I like to remind everyone who's not a plant scientist is that unlike humans and mammalians, plants don't have an adaptive immune system. So plants need to be ready at a germline level to make sure that they are able to fight off and fend against all possible attacks, which puts a big burden on the genetics of the plant because it has to be ready for all different aspects of an infection. And in the classical sense, what we understand as plant immunity is divided into these three layers called pattern-triggered immunity or PTI, effector-triggered immunity or ETI, and the part that I am mostly interested is between the PTI and ETI pathways, where pathogen-secreted effectors target different intracellular components and trafficking pathways to dampen immunity. Right? And because this is the most important aspect where the pathogens try to gain an upper hand over plant immunity, the pathogens have evolved to have a lot more effectors. Pathogens have evolved to have a lot more effectors that target various different pathways. And in some cases, a lot of them are redundant to make sure that if one effector is not able to dampen a pathway, that is a backup plan. Right? And based on this, we designed a hypothesis, which simply we named as many pathways to defense. And this is part of a bioessays article that I authored. And if anyone has not done that, and if they get an opportunity, because it's an invitation only thing, I would strongly recommend you to take this opportunity because it allows you to explore your hypothesis without actually having solid data. It allows you to write down all possible hypotheses and make a case of why you favor one hypothesis over the other. In this case, what we did was to propose a hypothesis which says that there are many pathways to defense. And each pathway depends on what components are there. For example, there's a microtubule-dependent pathway or an actin-dependent pathway, and then there is a unconventional protein secretion-dependent immune pathway that allows secretion of immune proteins or immune cargo, not only to the apoplastic space, but in the case of fungal infection, also to the hostorium. Right? Now, the interesting and unknown thing about this pathway is that the interaction and redundancies between these pathways are not known. For example, if one breaks down the microtubule pathway, how much of the burden is taken over by the actin pathway, or are there more open pathways opened to ensure that secretion to the apoplast or secretion within the cell remains constant? Right? And one of the things that I'm interested in is to elucidate the redundancies or the interaction between these pathways. And in doing so, one of the pathways that we have identified is the TNAP1 dependent pathway. So in a forward genetic screen, we identified TNAP1 as being required for both exocytic and endocytic trafficking, and that plants that lack TNAP1 had problems with bulk secretion. And TNAP1 manages to do that by having a very unstructured C-terminal domain, allowing its N-terminal domain to interact with the microtubules. So in a sense, TNAP1 acts as a bridge that connects microtubules with post-Golgi vesicles, enabling faster and more directional transport. So when we knocked out plants and tested them against the bacterial pathogen Pseudomonas syringae, what we found was that a TNAP1 mutant was more susceptible compared to the wild-type Columbia, and complementing a TNAP mutant with a wild-type TNAP1 restored the resistance phenotype. While it is important to note that TNAP1 is more susceptible compared to Columbia, it is nowhere as susceptible as a hypersusceptible EDS1 mutant, suggesting that there is more going on in the TNAP1 mutant than just susceptibility. And this is true against also a filamentous pathogen like the oomycete hyaloperonospora, where TNAP1 is also more susceptible compared to Columbia. So once we established that TNAP1 is important for resistance against biotrophic pathogens, we tested if major pathways of biotrophic resistance are affected in TNAP1. And what we found during a transcriptomic analysis is that upon infection, TNAP1 mutant, which is more susceptible, is transcriptionally comparable to wild-type Columbia, which is to say in a single line, is that the canonical defense pathways that are activated in Columbia and not activated in a hypersusceptible immune mutant like EDS1 or SID2 is not affected because of the loss of TNAP1. That is, all the pathways that are required for activating resistance are fully functional in a TNAP1 mutant. Once we established that, we wanted to check if the TNAP1 pathway is additive to the canonical salicylic acid pathway, which is important for defense against biotrophic pathogens. But before making higher order mutants, we wanted to ensure that salicylic acid indeed is produced. And what we see here is that post-infection with DC3000, the free salicylic acid levels in the TNAP1 mutant is comparable to wild type, whereas in the biosynthetic mutant, which acted as our negative control, there is very low salicylic acid. So seeing that the TNAP1 mutant is able to make salicylic acid, we generated a double mutant of TNAP1. And as you can see, this is bacterial growth against the different mutants on a log scale. The TNAP1 mutant and SID2 mutant, which is the biosynthetic mutant for salicylic acid, are both more susceptible than Columbia. But a TNAP1 SID2 double mutant is more hypersusceptible, suggesting that TNAP1 and SID2 act in additive pathways or at least are downstream of one another. So then we tried to check if we pre-activate immunity, are we able to retrieve some of the susceptible phenotype? And when we pre-activated immunity by providing an SA analog BTH to the plants, pre-treated them before treating them with the Pseudomonas syringae pathogen, we found that both Columbia and TNAP1 plants were more resistant compared to the mock-treated plants, suggesting that given pre-activation of immunity, TNAP1 is able to activate immunity. But even this activation is not sufficient to alleviate the loss of TNAP1 mutant, and the plants are still more susceptible compared to the same treated Columbia plants. So checking that the canonical defense pathways are comparable and everything was different, we checked if TNAP1 is able to produce enough PR1. And what we found was that compared to mock-treated plants, a TNAP1 mutant was able to produce highly sufficient PR1 at an mRNA level. So we tested if this is because PR1 is an apoplast, works in the apoplast, it needs to be transported from the cell, from within the cell to outside the cell in the apoplast, if TNAP1 is affected in this. And to test this, we tested both the total PR1 fraction and the apoplastic PR1 fraction. And what you see post-infection is that in the total protein, there is no difference in PR1 accumulation in wild-type Columbia, a TNAP1 mutant, and a TNAP1 complementing line. But once you see the apoplastic fraction, you see that almost 80% of PR1 is not transported to the apoplast in the TNAP1 mutant. And the same is true for a PR2 mutant, suggesting that TNAP1 plays a critical role in exocytic secretion of antimicrobial proteins. Now, to test if this is specific only to the pathogen, or if every secretion during in the loss of TNAP1 is affected, we did apoplastic transcript proteomic analysis. And what you see on this figure here is the separation of proteins that are less abundant in the apoplast in the TNAP1 mutant versus those that are more abundant in Columbia. And on the left, you see plants that are treated with the bacterial pathogen DC3000. 7 different proteins were less abundant in the TNAP1 mutant compared to Columbia. And all of them, all the 7, belong to antimicrobial set of proteins, including PR1, PR2, the aspartic proteases SAP1, SAP2, suggesting that upon immune activation, TNAP1 is involved in specific transportation of immune cargo. An interesting aspect of this research that we are still exploring is a bunch of proteins that are more abundant in the TNAP1 mutant compared to Columbia, suggesting that TNAP1 also might be involved in either endocytosis of these pathways or endocytic pathway of these proteins, or maybe other pathways that are in balance with TNAP1 are now in an imbalance because of the loss of TNAP1, and therefore there is more secretion of these proteins. Our future research will focus on trying to understand if some of these proteins are also important for balancing out the loss of TNAP1. Now, once establishing that TNAP1 is important for secretion, we looked at other TGN proteins that are involved in immunity, and one of the most important proteins that is very well studied is Min7. Min7 is also located at the TGN, and it's important for both PTI, basal, and ETI defenses. So we generated a TNAP1 Min7 double mutant, and the double mutant was more susceptible compared to either the single Min7 mutant or the TNAP1, suggesting that TNAP1 and Min7 operate in different pathways. Now, to test if TNAP1 and Min7 co-localize, because TGN's trans-Golgi vesicles are highly heterogeneous, we tested if Min7 and TNAP1 co-localize along with at the TGN. So we used a TGN marker CYP61, and what we found was that 80% of TNAP1 decorated vesicles were also found to be decorated with Min7, which is a long way of saying that TNAP1 and Min7 are largely found on the same vesicle, but there are instances of some vesicles where you find only Min7 or only TNAP1, suggesting that there are also a small subpopulation of vesicles are TNAP1 specific or Min7 specific. So we came up with this model where, which is what I'm working on right now, is to understand the TGN as a hub for immunity, and also a very important hub for pathogens to target to dampen immune-related secretion. What has been shown by another lab is that Min7 co-localizes with HLB1, which is a protein that associates with actin. And our study shows that Min7 co-localizes with TNAP1, which is a protein that is associated with microtubules, suggesting that the microtubule and the actin cytoskeleton network have points of convergence. And the hypothesis that we favor the most right now is depending on what sort of pathogen and pathogen effector the plants confront. And if HLB1 dependent pathways are dampened, then microtubule and TNAP1 dependent pathways take the forefront of taking care of antimicrobial protein secretion. And if microtubules are dampened, then actin dependent pathways work. What would be interesting, and it's still not known, is what is the interaction between these pathways, how much of a workload is added in the absence of one of the pathways being affected. And from an evolutionary perspective, it is even more important to understand why would plants want to create such a huge hub of susceptibility, because HLB1, Min7, TNAP1 co-localize to the same TGN vesicle, suggesting that most immune-related cargo goes not in a vesicle-dependent manner, but vesicles are packed with immune-related cargo and then transported based on the pathogen infection. And this allows a window of opportunity for the pathogen to target specific vesicles. And this would be a very interesting field to study in the field of plant pathogen coevolution. So to sum up, I hope that I have shown and convinced you all about the importance of TNAP1 and immunity. And the working model that we have is that in the presence of TNAP1, the secretion of antimicrobial proteins to the apoplast is conducted at a very high and directional speed. And in the absence of TNAP1, because TNAP1 is important for the directionality and efficiency of secretion, the secretion of antimicrobial proteins is much reduced. Although there is still some secretion of PR1, suggesting that there are alternative pathways that are still functional in the absence of TNAP1. Right? So, but the most important thing is that TNAP1 operates in a non-canonical defense pathways, allowing us to engineer more resistance. We are the first ones to show that microtubule binding TNAP1 is able to bridge post-Golgi vesicles for specific cargo transport. and that massive massive secretion is important for reducing pathogen growth. Right? What we're trying to do now is to engineer this and tip the balance more towards defense without compromising growth by leveraging the functional importance of TNAP1 in a non-immune canonical pathway, which means that the classical sense of defense versus growth trade-off is that if plants invest more energy in defense, they have less resources to go towards growth. And that's why we see most proteins or engineering aspects of trying to engineer more resistance, which increase salicylic acid, result in stunted growth. What we have with TNAP1 here is an opportunity to not affect the salicylic acid pathway, but just enhance the speed of things going to the apoplast. In a sense, it's like building better highways, which will enable faster transport of the proteins without using massive resources. With this, I would like to acknowledge my whole lab, especially Professor Brandisi, who's moved out of her expertise and helped me set up a plant microbe interaction aspect in our lab. We are in active collaboration with Shenyang He, and a lot of Min7 work was done by Kinya Nomura. And we are also doing a bit of crystallization with Soichi Wakatsuki's lab at Stanford, along with Dara Daulat Shai. And most importantly, I'd like to acknowledge and thank the plant postdocs for this opportunity and also connecting me with my mentor, Rebecca Roston, who's been very helpful. With this, I'm ready to take questions, if any.
Thank you very much, Dr. Deepak. Very nice presentation. Thank you for sharing all those insights about plants and microbes. I'm watching the chat. So far, we don't have any questions, but let's give people 1 or 2 minutes to think. And while we wait, I'll just take the opportunity to ask you a question. Maybe could you tell us a little bit more about the broader implications of your findings, and how do you anticipate the next steps to bridge this knowledge with applications in plant science?
Sure. So the broadest implication that I can think of is helping this as a translational mechanism. So think of it this way. You produce a lot of antimicrobial proteins, but the plants are limited in how much of it can be transported fast, right? And by overexpressing TNAP1, for which we have data, we can show that plants are more resistant without having a trade-off in growth or shorter things. So I think one of the broader implications is this could be used as one of the tools to engineer better resistance without activating immunity, right? And the other broader implication is to bring back research onto the secretory aspects, because the logistics of defense or understanding how things move from one place to another has been an overlooked aspect in plant immunity. A lot of plant immunity has been in trying to focus on what are the receptors, what are the NLR proteins, or what effectors work. But there are certain pathways which are sort of bread and butter. Right? They're not as mechanistic in terms of A interacts with B, causes C. What we identify now is that if you affect microtubules, you affect secretion and therefore affect resistance, right? And which also is evidence with the importance of how many effectors actually target actin and microtubules compared to how many effectors that target transcription factors or NLR proteins. About 30 to 40% of the known effectors, at least, target cytoskeleton in one form or other. But that, I think, is the broadest implication.
Thank you. Okay, so we don't have questions yet, but you are welcome to write your questions to the chat, and we can communicate later on with our speakers. So we're going to move on to our next talk right now. Next, we have Dr. Renee Dale. She is a postdoctoral fellow at the Donald Danforth Plant Science Center, and her research uses experiments and mathematical modeling to study developmental processes across scales. With this research, she aims at understanding the mechanisms underlying plant adaptation. And today, she's going to talk about mathematical modeling for high throughput phenotyping. So Dr. Dale, please feel free to share your screen, and it's all yours.
Thank you for the opportunity to share with you some of my work today. And I'm very honored to be a part of this symposium. So I'm going to talk to you a bit about my work modeling Cetaria for high throughput phenotyping. Oh, okay. So there are more corn plants in the Midwest than there are stars in the Milky Way galaxy. But as drought and other environmental stressors continue to increase, corn production is threatened. And what I'm interested in is understanding what enables plants to be resilient to stress. At the Donald Danforth Plant Science Center, we have this automated phenotyping platform that lets us measure large amounts of plants in a dynamic fashion. So every day you get an image of the plant, then you can extract phenotypic traits from that image. You can also weigh and replace water in the plant. That also lets you track how much water the plant is using. So as the plant grows larger, it gets more and more water as it uses more water. So what this lets us do is it lets us leverage high throughput data, such as diversity panels, so that we can get the genetic diversity necessary to get at mechanisms underlying things like drought stress resilience. So when thinking about plant growth and water usage, while it's nice that this system allows us to track water usage, there's also a bit of a positive feedback loop going on. So if you can imagine a plant that is larger, maybe it's larger so it gets more water. And this may be, more or less water use efficient. It's a bit hard to tell because of that positive feedback loop. So what I want to do is develop a model that can help us separate plant growth from water use efficiency so that we can get at the genetic basis for water use efficiency. So to do this, I have a couple of constraints because I want my model to work with this high throughput data. So we're limited in terms of what we're able to collect. So I want my model to capture growth and water usage, but I don't want it to have too many parameters so that I can get good predictions. So I'm working with Cetaria, which is it's a crop plant for millet, and also it's a model organism for things like corn. And the way it grows is it has a series of developmental units called phytomers. Each phytomer has an internode connected to a leaf. They're stacked up on top of each other. And at the end, Cetaria puts out its reproductive phytomer, which supports the panicle. So when I was developing this model, I needed a way to try to simplify growth so that I, again, can keep the number of parameters low. So what I ended up doing was kind of leveraging this semi-sequential growth to construct the model. So as I mentioned, you have these phytomers. They grow semi-sequentially. So first one phytomer emerges, then the next one, it grows for a certain amount of time, and so on and so forth until you shift to reproductive growth. So I tried to take this and translate it into a model. So thinking about it in this way helped me cut down the number of parameters. So in the model, we have the parameter for waiting time, tau, which is the emergence rate of the phytomers. There's also a grow time beta that describes how long each phytomer component or tissue type grows for. So in the model, we have reproductive growth, leaf, internode, and sheath growth. And so those parameters describe which phytomers are growing when. And then from there, I assume that allocation of resources is spread across whatever is growing at that time. So for example, with the first time point with the red line, we have just one phytomer growing. So that phytomer is going to get all of the resources available at that time. With the second time point, we have 3 vegetative phytomers and 1 reproductive phytomer. So it's going to be divided up between them. So the model also has some allocation parameters, such as the reproductive investment parameter, to describe how the resources are divided up. So here's an overview of everything in the model. So first we start out with, okay, so first we start out with biomass production. So this is, I assume that the amount of biomass available to be allocated at different points in time is proportional to overall plant size. Oops. Okay. That biomass then gets allocated, and there is an allocation fraction parameter for each of the tissue types in the model. And then we have the resource allocation dynamics, which describes which phytomer is growing when. And then at the end of the simulation, we can sum those allocations up and we get the accumulated amounts for each of the tissue types. So we get leaf length and internode length, and we also get composite traits like height, and we also have this feedback loop into the biomass production as the plant gets larger. After developing this model, I needed to collect some data to validate, to make sure that this model is not too simple and that it's able to capture the growth that we're interested in. So I did some experiments in the greenhouse, really just focusing on measuring the model parameters. So I measured leaf emergence and the grow time or the elongation time for the leaves and the internodes. I tracked the overall plant growth. And at the end of the experiment, I dissected the plants to get the final length of the leaves, sheaths, and internodes. So I found that the model is able to capture the growth of the system, and I used 2 very different genotypes to test the model. So the difference between the 2 genotypes is some of the parameters are different. That describes their different growth behavior. So on the left, I have wild Ceteria. This is a smaller plant and it's also very bushy. And on the right, I have the domesticated Ceteria. This one is much larger. And here I'm showing you a comparison between the model in orange and the data in blue for the internode lengths. And here are the sheath lengths and the leaf lengths. So overall, the model is capturing the behavior of the system very well. So I can say that it's not too simple. So moving forward, I'm ready to apply it to this high throughput context. Oops. Okay. Now that we're getting ready to apply it to this phenotyper data, we can add in that information about water usage and water use efficiency. And once I add in the water use efficiency, that makes the model fully contained, because we now have information about growth and water usage. So we kind of complete this positive feedback loop. So far we have many of the big pieces of the model have data that will inform those pieces of the model. And because I'm really focusing on predicting water use efficiency, I would like as much of the model to have data supporting it as possible so that I can improve the quality of my predictions. So coming off of the automated phenotyping system, we get water usage, height, and biomass. So far, at this point, we don't have any information that can help inform the resource allocation dynamics. or the relative allocation. So we're looking for leaf emergence, elongation duration, the number of phytomers, and panicle emergence would help us figure that out. So to approach this, I started working with the plant CV team at the Danforth Center, in particular Haley. And in order to get at emergence and elongation, we needed to do leaf identity tracking. So we were able to get that done pretty well. So now we have some of those pieces handled. And you might have noticed things kind of start to fall apart near the end of that video. And that's because we start to tiller. And on the left, we have, this is B100. So this is the domesticated Soteria. It typically just makes one tiller, maybe two. And on the right, we have the wild Soteria. This one is very bushy. So this causes some challenges for the model because the model is really kind of for the culm and the tillers, grow similarly, but there is a temporal delay. So it also causes some difficulty in terms of interpreting the data when we think about tracking plant growth over time. So to give you an example of this, here I'm showing you some of the biomass over time. This is using the images of the plants on the automated phenotyping system. And if I asked you how different you might think these two genotypes are, each genotype has a different color, you might think they should not be that different because they both are looking like exponential growth. They both look like about the same size, but they're actually those two very different genotypes. And the reason why they look similar is because of the tillering. So even here in this case, the A10, the wild Soteria, this one already has a panicle, so the culm has finished growing, and you're really seeing tillering to get that exponential growth. So to approach this, to try to improve the model, to measure and model this growth, we decided to extend this leaf tracking that we were already doing to look for when tillering begins. So here I'm showing you the leaf counts over time for the two genotypes. And already there's a big separation between the two, as we would expect. And we can also see when tillering starts. So typically 0 to 1 leaf will emerge per day. And at some point when tillering starts, you see 2, 3, or even 4 leaves emerge at the same time. So that tells us that tillering has started. And we also get an idea of how much tillering happens by looking at the rate of leaf emergence. And then what we can use that information for with the model is we can separate the growth of the culm and the tillers from the overall biomass that we're seeing coming off of the images. So for example, here we have the overall biomass, which is the sum of the culm growth and the growth of the tillers. So with the model, we'll be able to model the tiller growth separately because we will have an estimate of when tillering starts and how many tillers are then going to emerge after that. So going back to the model, we now have a new piece where we are able to separate the culm growth from the tillers to be able to model the whole plant. And at this point, again, we have several of the large pieces of the model are already informed. In terms of the resource allocation dynamics component of the model, we now have estimates of leaf emergence and elongation duration. We don't have a great way to estimate the total number of phytomers or the panicle emergence. The panicle, we're still working on trying to detect the panicle. It seems quite difficult, especially because the genotypes that typically put out a panicle are small and bushy and floppy. But because we have so much other information to fill out the model, this is something the model will be able to predict as well. And we have the tiller start time and the tiller rate to help inform modeling of the tillers as well. We still don't have any information on the relative allocation to the tissue types in the model. And this is going to have to be something the model predicts, because in order to get this, you'd have to do dissections, which is not going to be done in high throughput. So moving forward, we have extracted data from these images in high throughput to inform the model. So now we will be able to use the model to make predictions about phenotypes, in particular water use efficiency. The model will also make predictions about different dynamics in terms of resource allocation, and it will also predict things like bushiness by integrating all this information together. We'll be taking those predictions and looking for genetic loci underlying water use efficiency, and also the other phenotypes that we would be collecting thanks to the model. And going forward, I think it would be very interesting to expand this model to capture the growth of other plants that grow in similar ways with phytomers or nodes, like soybean and corn. And these also have different shifts to reproductive growth, and they shift to reproductive growth different numbers of times, which I think would be very interesting with the model. So thank you so much for listening to my talk. I thank my mentors and my collaborators. If you're interested more in the model development and the model validation part, I have the preprint out from last year, and hopefully the rest of it will be out this year. Thank you.
Thank you so much, Dr. Dale. I'm again looking at the chat, waiting for questions. If you have questions, please, you can use the chat to share that with us. And while we wait a little bit, I will also ask you a simple question. So did you have any unexpected results from these experiments? I don't do many experiments myself, but I always think that, you know, something unexpected might happen, especially when you do time lapse with plants. I guess, you know, you might discover some things. Do you have any experience with that?
Well, I haven't. I can tell you something that was unexpected from the greenhouse experiment I did for getting the model parameters. Yeah, so the grow times, I wasn't sure what to expect in terms of how complex or variable you might see, say, how long each leaf grows for, especially with different numbers of phytomers. But it turned out that it's just a multiple of when that leaf starts growing. So there's a really nice linear relationship between when you start growing and when you stop growing. So that was very exciting as a modeler, because that means I just have one parameter. Yeah.
So we have one question from the chat. Do you plan to get phenotypic data from the field to use in this model?
Yeah, I think that's also another thing that would be very exciting to do. And I have been working with some of the published data where they have, you know, those kinds of yeah, that kind of field-based data. Yeah, I think that would be a really exciting direction to go in.
I have one more question.
Yeah.
So as you are interested in determining the water use efficiency, so did you monitor the photosynthetic efficiency and the gas exchange rate in your plant system?
Yeah, that's a good question. We didn't, because it's just the images, really. Yeah, so that's.
But can you define the concentration of the environment, the gaseous composition in the chamber? What you're
using? This one, they have droughted plants and non-droughted plants all together in one big room. Yeah.
Okay, thank you.
All right, thank you so much again, Dr. Dale. We are going to move on to our next speaker. So now I want to introduce Dr. Sombir Rao. He's a research associate at Cornell University, go Big Red. As a scientist, He is interested in regulatory controls of carotenoid accumulation in plants and developing novel strategies for crop improvement. And today his talk is titled Unveiling the Treasure, Harnessing the Power of Carotenoid Metabolites to Improve Key Agronomic Traits. So Dr. Rao, please take on the screen. Feel free to project now.
So thank you, Israel, for the wonderful introduction. I would like to thank the organizers for allowing me to share my research in this very platform. So at Cornell, what we are doing is, so our lab works on the regulatory aspects of carotenoid metabolism, and I am specifically interested in to tap this underutilized power of carotenoid metabolites and use them to improve the agronomic traits of the crop plants. So my specific focus is on improving the yield, second, the stress resilience to the high temperature, and third, if I can enrich these crops with the nutrients, that is carotenoids. Why I am focusing on the yield specifically? Because I have a question that can we feed this growing population that's going to touch the mark 9.6 billion in the coming years? And the global yield production trend shows that there is going to be huge difference between the crop what we need to feed that much population and the production we are having in actual terms. So in all the staple crops including maize, rice and wheat, you can see there is a huge difference in the amount of production we need to feed the population and the production which we are actually having. So the studies demands that we should or we must double the amount of production what we are having in the current scenario. Furthermore, the abiotic stresses including the climate change is further adding to the problem, and it is also predicted that the climate change is going to reduce the crop production significantly. Furthermore, when we are like talking about enhanced yield or the food sustainability, we should also focus on that what we are producing in actual terms, whether our agriculture products are good enough to feed our nutritional requirement, what we actually need. So here is the data in 21st century when the scientists are like capable of exploring the outer space, we still have red spots in the all over the globe that shows that we still have a malnutrition, and that is associated with the vitamin A deficiency. You can see here the red areas are showing that the population in these areas is having acute uh, clinical symptoms that are because of the lack of vitamin A in their food. So the vitamin A deficiency, because of this vitamin A deficiency, more than 40% children in Africa are prone to have the blindness. 140 to 250 million children are at risk of developing acute infections, and around 20 million pregnant women are at the risk. So even the US is a major consumer of meat products, but still around 25 to 30% of the US population can have vitamin A from plant sources. So now you can think of the vegetarian who are like eating only the plant products can have only one source of getting this provitamin A is from the plants only, because the plants are the source for carotenoids, and these carotenoids can be converted by the human system to the vitamin A. So that's why we should have carotenoids in our food, so that because our body is not capable of producing or synthesizing vitamin A at its own. That's why we are studying or we are interested in carotenoids or to enrich carotenoids in our food crops. So if we are talking about carotenoids, so what actually the carotenoids are? So these are the natural occurring pigments that can be synthesized almost plants or any other photosynthetic organisms, and they can have different colors. So carotenoids group of pigments can have around 1900 various kinds of chemicals, and the major classes are alpha carotene, beta carotene, lutein, zeaxanthin, and lycopene. Furthermore, apart from providing soothing colors towards our eye, carotenoids have a much to do in the plant. If you can see at the level of the organelle level in the chloroplast, carotenoids are helping the photosystems to capture the light energy, and they are acting as antenna molecules. In the cell system, they are acting as a signaling molecules, and all over at the plant level, they are known to regulate the growth and even the yield also. That's why I'm more interested in knowing how this size of carotenoid pool is regulated inside the plant. The study shows that the available pool size of the carotenoids can be monitored or can be regulated at three levels. One is biosynthesis, second is accumulation, third is the turnover. So my specific focus is on the enzyme here, GGPPS and PSY, that is phytoin synthase, that is known to be a key rate limiting enzyme in this process. So you can pretty much change what's going in there in this uh, towards this flux by changing this enzyme. So I
Okay,
want
okay, enjoy.
to know whether this is how this enzyme is regulated in the plant system. So that led us to discover a novel protein, that is Nudix hydrolase. So my this story is going to tell you how Nudix 23 is going to regulate the carotenoid metabolism in the plants. So first we started with how many Nudix genes are there in arabidopsis, and we via a genetic survey we found that there are 29 genes, and out of these 29, 6 were belonging to, or these were predicted to localize to the chloroplast. I am interested in the chloroplast localized Nudix proteins because carotenoids are synthesized in this organelle, the chloroplast. That's why I'm focusing on these green dotted proteins here. So the question is whether these 5 Nudix proteins have a potential to change the carotenoid production. So we have a very good system to monitor whether a particular protein is having a potential to change the carotenoid production or not. So this bacterial system, we have put all the enzymes that are required to produce carotenoids. And now if you put any other uh, protein inside this system, you can just tell, oh, it's going to monitor or it's going to change the production or not. So here you can see out of these 5, Nudex 23 is outstandingly enhancing the levels of lycopene that can be seen here from the dark red color, and that the same can be seen from the graph where we have measured the lycopene levels. You can see there's a significant enhancement in the lycopene production when we are expressing this Nudix 23 in the bacterial system. Then we are interested how the Nudix 23 is increasing the lycopene. So then we quantified the key proteins that are involved in the lycopene production, that are PSY and GGPPS. You can see as compared to the empty vector control and other Nudexics we are checking here, Nudex 23 is really enhancing the protein levels of PSY and GGPPS significantly. Furthermore, because our model of study is here is Arabidopsis, and we are focusing on the rosette leaves of Arabidopsis. So you can then we are interestingly like which one of the best, which Nudix is having the highest expression in the Arabidopsis. So here you can see Nudix 23 is outstanding and it is having very high expression, and it is also localized to the chloroplast. So these things, 3 things, yes, it is like enhancing the lycopene, modulating PSY levels, having high expression, and it is localized to the chloroplast, make Nudix 23 is the best protein to go further and to know how it's working. So okay, we know now Nudix 23 is modulating the PSY and GGPPS level in the bacteria, but how about plants? So to know that how Nudix 23 is behaving in the plant system, we generated the overexpression lines for Nudix 23 and the knockout lines, and we quantified whether Nudix 23 oil lines have a modulation in the protein levels of PSY. You can see here Nudix 23 overexpression lines have a really high amount of PSY and GGPPS as well, while the knockout has the lower levels of PSY and GGPPS, but there is a no change in the transcript levels. This data shows that Nudix 23 is only modulating the levels of PSY and GGPPS at protein levels, but not at the transcript. Furthermore, okay, now we know Nudix 23 is enhancing the level of PSY and GGPPS, but do it actually enhance the levels of carotenoid in the plant system? So to check that, we quantified the carotenoid levels in the Arabidopsis leaf, and we know that, and we found that yes, Nudex 23 oil lines is really having high amount of carotenoids as compared to the knockout lines. So this confirms that overexpression of Nudex 23 can enhance the levels of carotenoid in the plant system. So now the question is how Nudex 23 is enhancing the protein levels of PSY and GGPPS. One assumption is maybe Nudex 23 somehow physically interacting with PSY and GGPPS, and it's doing this function. So to prove that whether it is like interacting with PSY and GGPPS, we did Y2H BIFAC and co-IP analysis and found that yes, indeed Nudex 23 is physically interacting with both PSY and GGPPS in both in vitro and in vivo assays. Furthermore, we are interested to know how this interaction is modulating the levels of protein of PSY and GGPPS. So we are proposing that maybe Nudex 23 is somehow modulating the stability of PSY and GGPPS so that this protein can stay up to longer times in the plant to enhance the carotenoid biosynthesis. So to check that, we utilized a CHX cyclohexamide method where we treated the wild type overexpression and knockout plants with the cyclohexamide, and so what the cyclohexamide is going to do, it's going to block the fresh synthesis of proteins. Now there will be no more new synthesis of proteins, and you can monitor what's going in with the already synthesized proteins. So here I'm monitoring the levels of PSY and GGPPS after different time points of CHX treatment. When I measured at 9 hours, you can see at 9 hours both the PSY and GGPP proteins were not there in the wild type, while in the oil lines where Nudex is in much there, the PSY and GGPPS proteins sustained for the longer time, even up to 9 hours, while the turnover is really fast in the Nudex knockout lines. So you can see here Nudex 23 orexin plants really have high stability or or slow turnover rate for this PSY and GGPPS. So this slide confirms that yes, Nudex 23 is affecting the stability of PSY and GGPPS, and they are keeping the PSY and GGPP for the longer time in the cellular system. Furthermore, there are studies that shows that if PSY and GGPPS are in a complex, this complex is highly efficient in channelizing all the available precursor that has been used by different pathways to synthesize different things. So if these PSY and GGPS are together, they can actively channelize all the precursors specifically towards this pathway so that you can have more carotenoids. So to check that whether this Nudex 23 is forming a complex of PSY and GGPS that is acting as a metabolome to channelize all the precursor to this pathway to attain a high carotenoid synthesis state, we confirm this by a BN page analysis, where we confirm that you can see both Nudex 23, PSY and GGPPS are in the same size range. That confirms that these three are forming a complex, and this complex is acting as a metabolome that is actively channelizing all the precursor towards the carotenoid biosynthesis, and that's why the plant is having high amount of carotenoids. It can be seen from this model that GGPPS and PSY are like brought together by Nudex 23. This form a complex, and this complex is actively channelizing this substrate that can be utilized by various pathway towards specifically this pathway, so that you can have high carotenoids and plant can achieve a state of high carotenoid biosynthesis. Furthermore, you can see in this pathway, carotenoids are like continuously degraded towards the signaling molecules, or you can say the derivatives that we call them epicarotenoids. So this research area is really emerging research areas. It's less than a decade when these chemicals has been discovered. So now I am like interested in knowing the potential of these epicarotenoids, how these can change the agronomic traits of the crops. So I checked the mutant of a carotenoid that is having really less amount of carotenoids in the stamens and in the flower. So we found that these mutants are having really low amount of beta carotene specifically. So now I want to check how the beta carotene is affecting the fertility of this plant. So you can see that the plant which is having less beta carotene or carotenoids, or the mutant which is having less beta carotene, is really having poor pollen germination, while the wild type is really good in germination. Same is with the tube length. The mutants that are compromised in apocarotenoid production, or you can say beta carotene production, is having less or reduced length of pollen tube. That same can be seen from the graph here. The PSY is having mutant is having really less amount of germination and a shorter tube. Furthermore, we want to check whether if you what about the supplementing of these mutants with the beta carotene. So if you supplement the pollens of this mutant with the beta carotene, it can significantly enhance the germination. So you can see here only beta carotene can improve the germination and the tube length, but not the other carotenoid, that is lutein. Furthermore, we are interested in knowing how it's happening. So we just checked, because rose reactive oxygen species are known to affect the pollens very badly. So we are just want to know what's happening with the rose in there when we are checking the pollens of mutant. So you can say, as compared to the wild type, PSY mutant really accumulates very high amount of rose. That makes sense to me, because carotenoids can act as an antioxidant system. So in wild type, we have a high amount of carotenoids. So that's why they are just squenching the excess amount of rose, but PSY is not capable of squenching rose. That's why it is really having high amount of rose. Same can be seen in the tube, when the mutant is having really small length of the tube, because of the high amount of reactive oxygen species that are not letting the tube to grow further, because it is having a damaging effect in the pollens. So now the question is really beta carotene is involved in this system, or it can squench the rose? So we treated the wild type plant with the H2O2, that is a really potent reactive oxygen species, and you can see there is this reduction in the germination. But this germination can be recovered if you supplement this system with the beta carotene. You can say beta carotene can really squench the rose. Same can be seen here by the dab staining, that supplementing of these pollens with the beta carotene can really reduce the reactive oxygen species levels inside the pollen tubes. So now we are interested whether it's beta carotene only, or its derivatives or the apocarotenoids can also have the same effect. So if we treated, so to check that, we treated the beta the mutant pollens with the beta cyclosterol and beta inon. These are the two major classes of the apocarotenoid we are working on. So you can say the treating of the pollens with mutant pollens with beta CC and beta inon can really improve the germination of these pollens. So this confirms that yes, both beta carotene and apocarotenoids can improve the pollen tube growth or the germination. Furthermore, the third aspect I'm focusing is on the yield, whether these regulators of this pathway or proteins that has been discovered to regulate this pathway can be utilized to enhance the yield in tomato. So this our lab has discovered, or the lab is like working on this protein, orange protein, from I think 2 decades, more than 20 years, I will say. So this gene has been discovered by this lab, and they have shown that if you overexpress this gene in cauliflower or maybe melon or any other crop species, you can see how magnificent effect it can produce in terms of accumulating carotenoids. It can make everything orange. That's why it's named as orange. So now I'm interested in knowing whether this orange can also do the same thing as it's doing with the color here in the terms of yield. So we overexpress, to check that we overexpress this orange protein in the tomato lines. You can say they are beautifully increasing the levels of tomato fruit here in the oil lines. You can see while there's a significant reduction in the CRISPR-Cas lines for these lines in tomato. So that shows that yes, the OR can really double the amount of fruit produced by per plant, and it can really double the yield. Furthermore, we are interested whether it's the how tomato is going, how the OR is enhancing the yield. So in tomato, if you are saying enhancing yield, it can be you're increasing the size of the tomato, so that can have more weight. Or the second is you can only increase the number of fruit producing per plant without effect on size. So you can see here we found that OR is significantly enhancing the number, while there is a no change or no off target effect or no off trade, you can say, on the size. So the size remains the same, while there's a huge difference in the number of fruits producing in these overexpression lines. Same can be seen here in the knockout lines. There is a significant decrease in the number of fruits per plant. Furthermore, we want to know how this OR is increasing the number of fruits in the plant. So we check that in each truss where the M82, that so we use two different backgrounds. One is the M82, that is really very poor cultivar in terms of yield and pollen fitness. While we for the CRISPR lines we use the AC cultivar that is really having good yield. So we want to check the effect by silencing the OR in very good variety, and we want to see its pronounced effect when we are overexpressing in the bad variety, whether it can improve the bad variety or it can make the good variety worst. So you can see if we compare the number of fruits per truss in M82 and the oil lines, you can see the oil lines is having significant higher number of fruits as compared to the wild type. While in case of AC, that is known to have really good yield. If you silence specifically OR, it can just reduce the number of fruits from like 7 to 2 or 3 fruits per truss. So this shows that yes, OR is specifically modulating the number of fruits a plant can produce on each truss. Interestingly, what we found that the change in the number of fruits per truss is because the OR lines can improve the pollen fitness in these lines. So you can see the germination of pollens is really higher as compared to the MAT2 in OR or expression lines. Same is with the pollen tube length. While in contrast, the knockout lines of OR are really bad as compared to the AC in terms of both germination and tube length. So this shows that OR is modulating high fruit, or OR is setting high fruit because of the enhanced pollen fitness in the OR oil lines. Furthermore, to check what's happening in the plant system or in vivo, so we want to check whether it's also affecting in the plants growing in the greenhouse. So what we did was we just monitor how the pollens are growing the pollen tubes through the stigma to fertilize the oval to set the fruit. So if you just track the germination of the pollen tube in the stigma, you can say the in the OR oil lines, the more pollens are germinating, or more pollen tubes are penetrating the stigma to fertilize the ovule that can have more fruit. So you can see there are significantly higher number of pollen tubes growing through the stigma of these overexpression lines as compared to the MAT2. In contrast, if you compare the knockout plants with the wild type, they're really having very less number of pollen tubes growing through the stigma. So that confirms that yes, OR is indeed regulating the growth of pollen tubes in the plant system. Furthermore, we are interested whether it's the female part or it's the male part that is crucial for increasing the yield. So what we did is we just fertilized OR MAT2 ovary with the MAT2 pollens, and we found that this number of tubes are growing. But if we replace this MAT2 pollen with the OI pollen, you can say there's a significant increase in the number of pollen tubes growing in the stigma. That means OR pollens are specifically involved in here, while the female is not involved at all in contributing towards the high fruit number of this. Furthermore, to confirm that what's going in there, we also cross the AC, which is having really very good number of pollen tubes penetrating the stigma. If you just replace the pollens of AC here with the pollens of OR knockout lines, you hardly see any pollen tube growing there. So that also confirms that yes, pollen are the key here, that are the major determinant of yield in here. So now to focus how, or to pinpoint how OR is regulating, what target genes OR is regulating in doing so. We did a proteomic analysis at 2 stages. One is before fertilization and after fertilization. And we found that there are around 155 proteins that are upregulated in the OR oil lines at pre-anthesis stage, and 23 are at the post-anthesis stage. Furthermore, we just pinpoint by using geoanalysis and by literature search, we pinpoint 3 proteins that are upregulated in the oil lines while they were downregulated in the knockout lines. And we are specifically interested in the PIN4 because PIN4 is known to regulate fruit set in different crop species, and it is the protein that can act as a transporter of auxin, because auxin is a key player in fruit set. So that's why we are specifically focusing on PIN4 here. So we want to know whether OR is only affecting the levels of PIN4 at protein or the transcript also. So we check the transcript levels, but there's no difference in the transcript levels of PIN4 in the OI or in the knockout lines. So that confirms that OR is specifically acting at the post-translation level. So now we want to check whether they are physically interacting or not. So by yeast-2 hybrid, yeast-2 hybrid and BIFAC assay, they confirms that yes, PIN4 is interacting with OR. But what OR is doing, how the OR is increasing the levels of PIN4 protein. So we did a chaperonic assay, because OR is known to have a chaperone domain that can help proteins to fold properly after heat stress or after other point of other things. So we check whether OR can show any chaperone activity towards PIN4 or not. So yes, if we overexpress PIN4 with OR, it can really capable of squelching the pin4 protein after the heat stress treatment in the bacterial system. So this confirms that yes, OR is having some whole day's activity towards pin4. That's why it's maintaining its protein level higher in the plant system. Furthermore, now we are interesting if pin4 is really the good target or not. So the one thing we can do is we can just quantify the amount of yield or the pollen fitness in the pin4 mutants. So you can say in the pin4 lines, it is really the pollen fitness is really compromised and they have really low levels of pollen germination and the yield as well. So indeed, pin4 is a really good target because silencing of pin4 also have the same phenotype what we are getting from by silencing the OR. Furthermore, to confirm that the OR is enhancing this yield through the pin4, we did some genetic screens where we just overexpress the OR in the pin4. So pin4 is not in there. If OR is acting through pin4 in these lines, we should not have any change in the yield. So same, the orange color of the flowers can see yes, OR, the orange was really indeed was overexpressed, and if you overexpress OR, the fruits can become little bit of golden or orange or yellow kind of because they are accumulating really high amount of beta carotene. So these color confirms that OR is really overexpressed in the pin4 background. And if you compare the amount of yield in these lines, there is not much change in the total fruit or yield per plant or per truss. So this confirms that yes, indeed OR works via pin4. And here is the model in the OR where in the lines where OR is really high, it can stabilize pin4 by binding with it, and then it can set more fruits by increasing the pollen fitness. While in the case where OR is not there, the pin4 is going to degrade and is going to create a environment of low oxygen that is not good for pollen fitness, and it can have really low fruit set. So in conclusion, I have just told you like Nordic 23 can act as positive regulator of PSY and GCPS. Beta carotene and other carotenoid derivatives can enhance the pollen fertility, and OR is a good target to enhance the fruit yield in tomato. So I would like to thank the organizers and my lab members, specifically my wonderful mentor, Dr. Lee, and my wonderful undergraduates. They have been phenomenal in doing or helping me with all the stuff. Thank you very much.
All right, thank you so much, Dr. Rao. It seems you're ready to change the world from pigments to auxin and back. We are a little bit over time, so I'm just gonna run. There's a question in the chat. So, are there whole plant phenotypes associated with NUTX23 overexpression? Did you?
Yeah.
Yep.
So there's no change in the oil lines if you see the leaf, because in leaf the carotenoid production is always like balanced by the amount of chlorophyll is in there. So they really mask the carotenoids. You cannot see it. But in the knockout lines we have a phenotype. They are really slow in growth. So they have really uh, like delay in the flowering, and they are really growing very slow.
All right, thank you so much. It seems we don't have any more questions.
Sir,
Oh.
can I get a question in?
Go ahead.
So, Sombir, great talk. One question: with all the pollen phenotypes, how does the cytoskeleton look? That's what I'm interested in.
Well, yeah. So that's a good question actually. So when I'm treating the apocarotenoids with the pollens, so now I have the proteomic data of that tissue. So now I'm having several actin binding proteins. So my thing is I'm going to work on that, what you are asking now. So that's my plans. Yeah.
Okay, so if we don't have any more questions, I would like to thank again our speakers, Dr. Deepak, Dr. Renee Dale, and Dr. Sombir Rao. Congratulations again on your award. Thank you so much for attending our symposium today, and stay tuned for more activities and events from Plant Postdocs. Have a nice day and see you next time.